Maybe we'll get over 50 today, 50 people.
Let's wait 1 minute more.
Go get your friends, tell them to log on. We're going for 50. You don't know anything about birds? That's okay. Come learn.
To be fair,
Anyway.
I don't know anything about birds, so.
Me either. I work at the lab of ornithology. So know a few things about acoustics, but not so much about birds. We're getting there.
Right, exactly. Same boat.
I will try to contribute a little today.
So we may get started with this. Hello, everyone. And I'm Larissa Sugai. For those that I haven't had the chance to meet, I'm an extension associate in the Kelisa Young Center for Conservation Bioacoustics at the Cornell Lab of Ornithology, member of the capacity building team. And it's really a pleasure today to introduce you all to Cristian. And Cristian is going to be our speaker. Cristian, I met Cristian when I was going to Spain, and Cristian was I was moving from Brazil to Spain, and Cristian was moving from Spain to Brazil. He was going to do part of his postdoc there. So Cristian is an avian ecologist, and he's now at the Universidad de Alicante in Spain, and he has been doing some amazing research with many aspects of avian behavior using passive acoustic monitoring. And in particular, he has been developing methods to estimate vocal activity and density and abundance estimates of bird species, including some endangered species, both in Europe and South America, specifically in the Pantanal wetlands. So Cristian has just returned from a long summer trip, and even though he was super nice to accept the invitation for joining us here today, so Cristian, welcome, and I'll pass the word to you.
Okay, thank you, Larissa. Thank you to all of you. Good morning, good afternoon, good night. I don't know, but thank you for being there. So I will be talking today. Well, thank you also to the Cornell Lab for the invitation, of course. Sorry, I almost forgot. So I will have some noise in the background. Maybe you. Thank you. Someone needs to close the microphone. It's good now. So I will be talking about if it is possible to estimate bird density using passive acoustic monitoring. So this is the main question that we will try to solve today during the talk, and then probably we have some chat, some discussion. We can try to develop it a little more, because it's not the same if you are recording only one bird or if you are recording 2, 3, 4. Sometimes this is very interesting for monitoring programs, and you don't only want to know if the species is present or not how many there are, or how it's going, the population trend, etc. So at the very beginning, I will make a very short introduction. I promise no more than 3-5 minutes, because most of you are experts on the topic. So why using passive acoustic monitoring, and what is this technique? Then I will introduce the methods available for estimating bird density using passive acoustic monitoring. I will focus mainly to describe the methods included in that review that was published last year. But then I will explain further 2 of these methods that I have worked most, like the detected vocal activity rate and Q counting. So, well, what is passive acoustic monitoring? This is a trending technique that a lot of researchers, managers, and people are using today. So the technique, you need to deploy some acoustic devices in the field, usually programmed to record during periods of interest, usually up, down, it depends on the species. And it has been used a lot to monitor marine mammals, but also in the terrestrial environment for bats, birds, and anurans. So the main workflow usually that you have a target species that you are working with during your PhD, your postdoc, or some threatened species, whatever. And you need to know if the species is there or not, or what is happening. So you leave some recorders in the field following a protocol. So you always record during the same hours, during the same period of the year, etc., to allow comparisons. At the very end, you have a large acoustic data set composed of sound recordings, usually WAV files, that you need to analyze. And this is the first problem, because you can hear the recordings, but sometimes this is just impossible. Or you can use an automated software, but for most of the species, this is very costly, although there are some good machine learning approaches like BERNet that you from Cornell Lab are developing. Thank you for that. So usually using this software, you can identify the species if you have which different species you have there, but also the number of calls, of the number of vocalization. This is very important for most of the methods that we are talking later today. And then you need to assess this recognizer, if this is working well or not for detecting the species, if he's making a lot of mistakes, etc. And then at the end, you have your database and you can work with them to solve some ecological questions. So this passive acoustic monitoring has a large number of advantages. This is because of that, there are a lot of people using them. One of them is that you avoid the use of different researchers, not the personal biases, because usually if you have different people collecting data, they have different expertise, different hearing ability. So it's not easy or not fair to make comparison between people, between sites, years, etc. This is also a passive technique, so you can leave the recorders there and you move out, so you are not disturbing the natural vocal behavior of the species. This is especially important for some size species. It works very well in inhospitable areas that are difficult to access, that have low visibility, like tropical rainforest, but also if you are working with nocturnal wildlife. Even these recordings can be reanalyzed. So it can be reanalyzed looking for different species, but even you can try to solve different questions with the same species. So this is very useful. Sometimes with the same data set, you can make a lot of studies. And it allows researchers or managers to increase the scale, the spatiotemporal scale, because you can leave recorders in a lot of places and recording during the whole year. This is the kind of monitoring program that you cannot make using observers. But there are some disadvantages also to this method, of course. So one of them is that it requires a lot of effort in terms of time of money. At the beginning, you need to make a big investment to buy the recorders, because most of them, they are not very cheap, but you also have to buy the batteries, SD cards, to keep them, the microphone, maybe you have to change the microphone every year, etc. So it requires some money, and especially a lot of time and expertise to analyze the recordings. If you have to develop your own recognizer, it's something like an engineer thing that most of we feed biology, we are not able to do it. So this is quite difficult. But one of the main disadvantages for sure is that it's very difficult to know how many, I'm talking about birds, so I will talk about to know which is the bird density around recorders. So there are a lot of monitoring programs that is not enough to know if there is the species is present or not, in whether you need something else. And because of that, there are a lot of people that didn't use these techniques. So it's very difficult to estimate densities of birds or whatever or the taxa is, because most of the acoustic devices that we are using nowadays, they are equipped with omnidirectional microphones. So they are not recording in a focal location, or they are recording almost the whole soundscapes. So this is very difficult to locate animals, to know if they are singing more to your right, to your left. And it makes that bird density estimation is very challenging using this technique, but it's not impossible. So this is possible with accurate data and applying some of the methods that are already developed. For sure, there will be new methods coming in the following years, or maybe that one. But here I will be talking about the 8 methods, including that review that I published last year with my colleague Juan Traba in IBIS. But for sure, for those interested in these methods, I will tell that it's better to read first this review from Thiago Marquez in Biological Reviews in 2013. This is something more focused not only in birds, not in general, in animal population, but probably provide a better background. And if you are only interested in birds, maybe you can go directly to the review that I'm talking, but I think that's a good thing if you read first that review from Thiago Marquez. So in that review, we identified 8 methods that have been already employed for estimating bird density. There is one of them that I call it the sound pressure level. That probably is difficult to estimate bird density using only that method, but it is it's good to reduce biases between acoustic and human surveys. Then there are two methods that are based on the deployment of several microphones, like creating an array of microphones or recording in stereo. Then there are another 4 methods based on the analysis of sonograms, try to discriminate individuals. This is a method that is not included in the review, but I will put it here because I think that it's very promising. And then there are the paired acoustic sampling, the use of soundscape indices. And the last 2 methods that I will develop during more time, the vocal activity rate and Q counting. They are also very easy to understand and to apply. So maybe for some of you, they are good option for starting to estimate bird densities or make some analysis. So the first method, that is the sound pressure level, this is a method that is very good to reduce the biases between acoustic surveys and human surveys. So the main thing here is that the sound level, the level of the sound in a sound recording, declines with the distance. So if you are able to estimate the relationship between the sound level and the distance of vocalizing birds, that you can do it in the field, if you are able to annotate the distance of the bird that you are recording, or you can obtain this relationship using playbacks, it allows to estimate the distance between the bird to the recorder. So in this way, you can, at least you can know at which distance the bird is vocalizing. So this is a very good approach to estimate bird density, or at least this is something that you can apply in some of the methods that I will explain later. So it allows you to know at which distance the bird is vocalizing. So the birds that are vocalizing closer to the recorder, they are vocalizing louder, and the ones that they are very far to the recorder, you will see almost a weak signal. Then there are this method that is based on the deployment of an array of microphones. So it can be you can have only one recorder equipped with 2, 3, 4 microphones, or you can set up an array of as long as you want, so maybe 10 recorders, each one with 3, 4 microphones. So the reasoning here is that the time of arrival of the signal to each of these microphones is different. So the vocalization arrives faster to the microphones that are closer to the bird, and they arrive much later to the microphones that are placed farther to the bird. So in this way, you can, if you are seeing the 3, 4 sonograms of each microphone, it allows you to triangulate the location of the bird. And then there are another method that is based on the analysis of stereo recordings. So you may have some recorder with 2 channels that you're able to record in the left channel and the right channel. So some studies, what they did, they recorded in both with 2 microphones, and they hear the recordings. So they were watching the sonograms to the left and to the right. So from the beginning, they know if one bird was closer to the right or to the left of the recordings. So they know what. So they identify in which of the channels the bird was vocalizing. And then based on the sound level, they create some categories like low quality, medium quality, and high quality. So they were able to identify the bird was to the right or to the left, and it was close, at medium distance, or far to the recorder. So if they have, for example, one bird calling in the right close and another bird in the right too far, they counted like 2 different birds. In that case, this study was that one from Williams et al. They were counting Eurasian bitterns. So this is a type of bird that they are usually calling, booming during the night, and they are not moving too much. For sure, this method cannot be applied to some birds that they are vocalizing while flying. But in that case, they found a very good relationship between the number of bitterns that they count in the field and the number of bitterns that they identified on the stereo recordings using just this technique to making a fair separation between right and left. And then for each of the channels, they create 3 categories, and they estimated the number of birds as the number of vocalization on each of these 6 categories, 3 for each channel. And then you can do the same. You can, so there is another method that is called sonogram analysis. This is pretty similar to the one that I described for the stereo recordings. So there is one study that the authors, what they did, they were checking, they visually checking the sonograms, and they estimated bird density as the number of birds that they detected vocalizing at the same time. Of course, like they didn't find a good relationship between point counts and this technique, because this is not a good method, I would say, because you can have 5 birds in some area and they are never vocalizing simultaneously. Even it might be expected, that they have they tried not to overlay. So they tried in this way, but it didn't work too well. But then there is one study from Durek et al. Trying to estimate bird abundance of yellow rails. And what they did, they were also checking they first visually checking the recordings. So they estimate the minimum distance between calls of one individual. So they have some recordings of different yellow rails, and they saw that the minimum distance between syllables, between calls of the same yellow rail was always 0.17 seconds. So they considered this window, and they counted the number of birds in some recordings as the number of calls within that time window. So if you have only one bird in this window of 0.70 seconds, you will have only one call. That is the sample shown in the picture A. And then in picture B, they identified that there were 4 yellow rails vocalizing, because there were 4 calls within that time window. I don't know if it was clear or not. If not, maybe we come back then at the end. But the issue is that they... Okay, we'll continue. Sure, I cannot hear you very well, Marina, but maybe at the end. So they take into account which was the vocal behavior of the species. They identify the separation between calls of one individual. And if there were more than one call in this separation, it's because there were more than one individual vocalizing. And they also found out.
So I'm going to print, I'm coming.
And they also found some interesting relationship between the number of birds identified from sound recordings and the ones that they were truly vocalizing. Then we have this method that is the one that is based on the individual discrimination. So usually there are a lot of bird species that vocalization differ between the individuals, owing to differences in their settings or air sac structure, etc. So this is the same that if you are talking with one friend, you know if this is Peter or Robert because of how they talk. So the same is valid for birds. So there are tens of studies that have proved that that individuals can be identified, but all of these studies uses vocal recordings. And as far as I know, this technique tried to estimate the density of number of individuals on sound recordings based on the individual discrimination have never been applied using passive acoustic monitoring. But I think this is very promising, and I guess that someone will do in the recent future. So I'm opening a door here. So someone want to take it. Then we have this technique, this method that is called paired acoustic sampling. So this is a method that you need to perform point counts. Usually, the most typical survey for counting birds, that you stay in the same location for 5-10 minutes, counting number of species and number of calls. So you need to make this work at the same times that the recorders are recording. This is because of that it's called paired acoustic sampling. So the reason in here that both methods, human and recorders, they have the same number of species and the same number of individuals available to detect. So the difference found between acoustic recorders and human surveys is because of the different detection radius or detection probability of each method. So imagine if a human is detecting 10 vocalization of the European robin and the recorder is only detecting 8 vocalization. So we know that the recorder is missing 2 of these vocalization. No? So when you apply this method on several sites and several areas or moments of the year, you can estimate a correction factor. You can know how many calls the recorder are missing when compared to human surveys or point counts. It also allows to estimate bird density from all recordings, not only the ones that you are collecting now. So you can apply these correction factors to recording that you maybe have from 5 years ago, and you can estimate bird abundance and compare to the recordings that you're collecting now. But one of the problems of this technique is that you need to develop one correction factor for each of the species that you are working with. So this is very costly, and for some rare or size species, it can be impossible to get these correction factors. Then we have the use of soundscape indices. Probably some of you already know about them, but because they have been typically used to estimate bird diversity from sound recordings, to get like an index of bird diversity or wildlife diversity in general, frogs and uran, etc. So when you are using this for diversity, the idea is that the larger the diversity on an area, the larger is the acoustic complexity of the recordings. No, if you have more species vocalizing, the soundscape will be more saturated and you will have higher values. So the reasoning for the people that use this technique was the same. If you have a larger number of individuals of one species, you will have a higher amount of energy, or you will have a higher complexity on the recordings. So in the first study, it was made by Orben and colleagues, they assessed the relationship between the acoustic energy index. This is an index that measures the amount of energy within a band of frequency, the band of frequency that this species was vocalizing. So the idea was that if you have 2, 3, 4 individual vocalizing, this index, this amount of energy, they will be higher than when you have only 1 or 2 individuals vocalizing. So they assessed the relationship between the acoustic energy index and the breeding density of a storm petrel, and they found almost a significant relationship. So it was promising to apply in posterior studies. But then there was one that they assessed 2 different indices, but it was probably one of the two most commonly employed for estimating diversity, like acoustic complexity index and the biodiversity index. They assessed both with the breeding density of the man's seawater, and they found no relationship, and it was not even close. So we need more studies assessing the use of soundscape for estimating bird density. And then I will move into the to the vocal activity rate and the Q counting, the 2 methods that I said that I was going to explain a little more in detail, because I have worked most with them. So this method, the vocal activity rate, this is the method most commonly employed for estimating bird density. It has been already tested, I don't know, like 25, 30 times with different species. So there are a lot of studies using this index. So the idea is that the vocal activity of one species on sound recordings is density dependent. So the larger the number of individuals vocalizing, the larger the number of vocalizations that you detect. So you should be able to correlate this vocal activity rate with the bird density, and you could estimate the relationship between these 2 variables. So at the end, you have a graph like the one that we can see here, that you have the vocal activity rate, the mean number of vocalization per minute, and the number of, in that case, seawater nests. So once you know that relationship, you could place a recorder in a new site. And if you know the vocal activity rate, you can have an estimate of how many individuals there were around the recorder. So we tested this method, I think that for first time for terrestrial species, because this was used a lot for seawater, petrels, etc., but it was not typically used for terrestrial species. So we apply the method for the European V-term and for the DuPont slab. So in both cases, we work it like in around 30 sites. And during 30 minutes, we have one observer counting the number of birds moving around the recorder. And we have the recorder working at the same time. So we have a kind of also paired acoustic sampling. So we can assess the relationship between the number of birds counted by the observer and the number of localization on some recordings. And for the DuPont slab, we have a strong and positive relationship. It was the same for the European V-term. So, okay, we were very happy, like, oh, we have an index to assess the abundance of that species to work in the future. But then we very fast thought that bird vocal activity greatly vary among days. So if you are recording only 30 minutes, well, we get a strong relationship, but probably it's not a good idea to use one recording of 30 minutes to estimate the abundance of a species. And probably you need to record during most days, because maybe one of the days that you left the recording, it was raining, or there was a lot of wind, or maybe the male was fighting with another male, or was, I don't know, copulating with her female, something. So the idea here was if the relationships between vocal activity rate and bird density is reliable, or how we can make it more reliable. So we assess this relationship by recording during a number of monitoring days and using an average value of the vocal activity over this period. So in this way, you decrease the influence of unknown factors. You record in 5-10 days. For sure, the mean vocal activity rate that you are using is more accurate than if you are recording only one of these days. But the question here was, okay, but how long we need to record? Because it's not the same if we only need to record 5 days than 50 or during 5 months. So we assessed this by estimating the coefficient of variation of the vocal activity rate. And we consider that the vocal activity rate was quite reliable when the coefficient of variation was lower than 20%. This can be objective. We took this threshold based on some medical studies, because there are not too much using this in acoustic studies, but we saw for different areas of the DuPont slab that recording around 9 days, it was a good solution to have a vocal activity rate with low error. For sure, this is a limited solution. This is not perfect, but for sure, also this is much better than recording during a single day. And we found the same pattern for different species in the Brazilian Pantanal, for Niger, for some Crathis. So I thought that it was a good idea to leave the recordings during 9 days, but now we are working with a larger number set of species. You can see their names in the y-axis, and in the x-axis, it can be the name of recording days that you need to reach a vocal activity rate with an error lower than the 20% of coefficient of variation. So for some species like for chupotus and for some type of owls, you need to record like for 19-20 days if you want to have a reliable vocal activity rate. This is because this species, they don't vocalize every day, or maybe they change from where they are vocalizing. So some days they are vocalizing in a different post, they are not in the same area. While for some territorial species during the breeding season, like the European robin or the chaffinch, it is enough to record during 4 or 5 days to have a reliable vocal activity rate. So it will depend a lot of the species that you are working with. I will detail some studies that we have used this vocal activity rate to solve some ecological questions. So we use this vocal activity rate as an index of the abundance of the DuPont slab. So there were 2 areas that we performed habitat management actions, and we wanted to see which of these actions was better. And we saw that in one of them that we removed all type of trees, the vocal activity of the species increased from 1 year to the one after the action, while in the area that we only removed the pines, this relationship was not so strong. So We saw which one was better. It was better to remove all type of trees. This is because this kind of species live in a kind of steppes. They don't like trees. We also used this index to study the daily and the seasonal migration of the European bittern. So we can see in these graphs that in the right one, at which hours of the day there were more local activity rate of European bitters when they were migrating, sorry. And at the left, we can see the seasonal pattern of the species, where we can see that most of the bee-eaters migrate during the last week of August. This is now. Spain is now full of bee-eaters migrating. You can hear them everywhere. And in that article, we also assessed the relationship between the vocal activity rate and the number of observations uploaded to a scientific platform. It was not eBird, because in that area of Spain, there is a different one that is called Ornithocat, that is more used by the people. And we found also a strong and very significant relationship between the vocal activity rate and the number of observations uploaded. And then, for example, in that other article, we also saw the relationship between the vocal activity rate, so the number of vocalization of bee-eaters on recordings, and the number of bee-eaters hunting. So in that case, we only counted the number of bee-eaters that they were hunting honeybees, because we put the recorders in beehives. So we saw also strong significance between hunting pressure of bee-eaters and the vocal activity of the species. And it allowed us to assess the relationship between hunting pressure and the flying activity of honeybees. This is because beekeepers are usually complaining about that bee-eaters, they are killing the honeybees. So we need money, we need more studies, we need some, well, especially they are asking for money, of course. So in that study, we saw that, yes, they are right. If you have more bee-eaters, or when the vocal activity rate is higher, there was a lower flying activity of honeybees, although this relationship was also marked by the hours of the day. So during the first hour of the day, at 9, at 11 of the morning, there was a much strong relationship. So if there were bee-eaters at this time, honeybees, they were not flying. But probably at the end of the day, when it is very hot and honeybees are very active, they were not so afraid of the presence of bee-eaters. So some future studies, we want to see if they have an impact on the honey production. That probably is more important than just if they change the foraging activity of honeybees. But so for me, the main thing here is not trying to sell this study or the previous one, is to see that you can use this kind of index, like the vocal activity rate, as an index of bird density, to try to solve some new questions that probably are very difficult if you have to be in the field in so much time. So I'm going to check the hour only. Okay. So then we have this other method that is called the Q counting. So in that method is based, so you need to know how many vocalizations there are in one recording, similar to the vocal activity rate, but then you divide the number of vocalizations by the Q rate of the species. So if you imagine that you are working again with the European bittern, and you know that the European bittern is always vocalizing, I don't know, 5 vocalizations per minute. So if you have a recording and you have 10 vocalizations, you can divide this 10 vocalization by the Q rate of the species, so 10 by 5, and you will get a 2. So it means that you have 2 bee-eaters vocalizing around recorders. This is the idea. So a good thing here is that you don't need to collect complementary data. So if you know the Q rate of the species, when you have the recorder somewhere, you can already have some kind of index of the density of the species. But the problem is that you need to know very well what is the Q rate of the species. So you don't need complementary data, but you need to know what is the Q rate of the species that you are working with. And one problem that I didn't describe, well, this is related to the sound level method. No? So the sound, the vocalization of the species, the sound level declines with the distance. So if you are, so in that study, in that study, what we did was to analyze the relationship between the sound level of the DuPont's lab localization and bird distance to the recorder. For that, we used playbacks, and we get this relationship. This is the maximum amplitude of the DuPont's lab localization related to bird distance using playbacks. And we saw that every DuPont's lab localization uttered within a radius of 160 meters had a maximum amplitude higher than 144. Anyway, the important thing here is that we use this value, the 144, as a threshold, as a fixed radius of our recorder. So when we analyze the recordings, if we have some vocalization of the species with a maximum amplitude lower than 144, we remove this vocalization. So we were using a fixed radius of 160 meters around the recorder. And then what we did, we had the number of vocalization within that radius. We had the number of males counted by an observer within that radius of 160 meters. And we assessed the relationship between the number of males counted in the file and the number of males estimated using Q counting. But to estimate the Q rate of the species, we use 3 different methods. So we have some directional recordings, that is the one that is highlighted, like the best, like when we get the best relationship. So we have, I don't remember well, but maybe 50, 60 DuPont's lab makes recording in the study area during 3-5 minutes. Then we have some behavioral studies that this was carried out on a different area. Because of that, probably we didn't find a much better relationship. And then we also estimated the Q rate of the species using the recordings of Ladis to Xenocanto, but it was the worst method, probably because the recordings of Ladis in Xenocanto, they were so perfect, so people was only uploading a short piece of the vocal activity of the species, that when the species were vocalizing very highly, so it was a great bias for this method. And at the end, we also made a kind of assessment about how many individuals and how long you need to record to have an accurate Q rate. I will not extend too much on this, but we saw that if you can only record a few males, 5, for example, you need to record during a lot of time, during 25-30 minutes each of them, to have a reliable Q rate. But if you have a larger number of males, it would be better to record a larger number of males during less time. I'm sorry, this is very confusing, but the idea is that we made some kind of cost effectiveness assessment, and the conclusion though was that if you can, it's better to record a larger number of males during short periods than recording only 5-10 males during longer recordings. So I think that makes 40 minutes. I have some slides, but probably you can read most of them in the paper, because I only made it was a summary about the advantage and disadvantage of this method, but I think that's better if we leave more time for the discussion, because the table, you can read it on the review, so it was only some pros and cons of each method. So I will just say thanks to my colleagues, all of them that have contributed during these years, especially to Juan Traba and Gerard Bota. And now it's the time to, well, I have some last sentence here that I saw that excluding the vocal activity rate, for most of these methods, there have been only 1 or 2 assessments. So the thing here is that we need more studies, we need more people trying to use these methods, or of course, develop new ones. So I only want to encourage you to do this. And now I think that's a good time if we start the discussion or make some questions. I'm sorry because I have to go very fast. I know there are a lot of methods that I had to explain in 2 minutes, but I tried.
So we've got a lot of questions in the chat. Thank
Okay.
you again, Cristian, for a really comprehensive overview of some of the approaches that I think are showing promise and that seem relatively doable for people that are interested in getting some more information about the number of birds around their recorders. So why don't we go, I think there were some multiple questions about Q rates. And so just the last question in the chat, is there a difference between vocal activity rate and Q rate? Carly's heard them used interchangeably, but maybe there's a difference that you want to explain.
Yeah, they are quite related because probably the thing for both of them, you need to estimate the number of vocalizations on sound recordings. So this is like so for both methods, you need to count the number of vocalization of the species. But for the vocal activity rate, for this method that I call vocal activity rate, of course, there can be different names for them. I'm using the one that I put on the review just for keeping the same here in the discussion. But for this method, for the vocal activity rate, you need to estimate the relationship between bird abundance and the vocal activity. So once you have this relationship, you can draw a line, a regression line, and you collect recordings in a new site, and you can put the number of vocalization of the new recordings in your graph, and you can have an estimate of the abundance that probably is more or less correct in 95% of confidence interval, whatever. And for the Q rate, you need to know, for the Q counting, sorry, this is called Q counting, the method. For the Q counting, you need to know the Q rate of the species. So if you know how often the species vocalize, and you know how many vocalizations you collect during a minute, 5 minutes, whatever, you can divide the number of vocalization by the Q rate of the species, and you get the estimate of abundance. So they are quite related, but they are not totally the same.
So it sounds like for vocal activity rate, the two things you need to know are the total number of calls detected and the total number of animals present. And then for the Q count, you don't need to know the total number of animals present. You just need to know the Q rate and the total number of calls. So it's right.
So yeah, you are right. So for one method, for the vocal activity rate, you need to know the relationship between the vocal activity rate and the number of birds vocalizing in a number of sites, 20, 30, I don't know how many. And for the Q counting, you need to know the Q rate of the species. So depending on which data you have, you can use one or another.
Okay. And what would be, for anybody interested in doing this approach, I think there's a good amount of people that study birds on this presentation. What's a good workflow? You mentioned the target number of try and get at least this many individuals. And then is it manually reviewing recordings to estimate the vocal rates? Can you walk us through the workflow to do something like this?
So in my case, I only use the vocal activity rate and the Q counting. So I have to count the number of vocalization, and I always use the same software. I think that it was Sonoscope, because these studies were like, I don't know, 4 or 5 years ago. But you can use, so for example, now I'm using Kaleidoscope. I have made some small tests with BirdNet for some species, working very well, by the way, with the Eurasian bittern. But you can use whatever method, whatever software you know to use, and you need, in that case, for the Q rate or the vocal activity rate, for the Q counting, sorry, you need to count the number of vocalization. But for other methods, like the soundscape indices, you don't need to count anything. You only put the recorder in R or whatever, and you get an index. It will be the same for some other methods. And then if you are using the microphone arrays of stereo recordings, this is a different approach. You need to open, you are using an array of microphones. You need to have the sonograms of each of the microphone, annotate when it's starting the sound. So in this way, because of the time of arrival, you can triangulate the location of the bird. So it depends a lot of the method that you are applying. How do you need to approach your data?
Okay.
I'm seeing now one question now about if soundscapes reflect bird density. Well, there are only 2 studies. In one of them, they found a significant relationship, and it makes sense because they assess the relationship between the amount of energy with the number of birds. So for sure, the larger the number of birds, the larger the energy. It could make sense. I don't know if there is some moment that the soundscape might be saturated. I don't know. But then the other 2 methods, they didn't work, but they didn't approach the method well because they use this choose on the acoustic complexity index and the biodiversity index. But if they work it with the man's seawater, and imagine that that species is vocalizing between, I don't know, I'm talking loud now, but maybe imagine that they are vocalizing between 1 and 2 kilohertz, but they consider a much higher frequency, like from 1 to 8 kilohertz. So they were getting into this frequency a lot of non-target species. So the assessment that they did probably is not too valid, but I have to put it in the review, of course. It was not working, but I will say that no one has already assessed with quality if there is a relationship between the acoustic complexity index or the biodiversity index with bird abundance. I will say that are not the best methods. Probably, at least it depends on the species, because there are a lot of species that they have very, I don't know how to say that the vocalizations are pretty the same between individuals. So there will be not a high difference if there are 2, 3, 4 individuals. It will depend on there are more or less variation between individuals.
Yeah, so we're getting a few different questions about the temporal aspect. So what if, I guess, Q rates, what if they change over the course of a day, or if they change over the course of the year? And how do you deal with this temporal component?
Really, I don't know. Yeah, for sure.
But you need to know.
Yeah, no, I was almost joking, but of course this is happening. So when I was talking here about the DuPont slur, I never said, but I was always talking about the breeding period between April and May. So if you apply this method for winter, it will not be a good approach. So you need to know which time of the year you are working. There will be some species that they just don't vocalize during winter, so it makes no sense to record them. So yes, it will be a problem for sure. And then there are another problem for the vocal activity rate, that I say that you need to know the relationship between bird abundance and the vocal activity in a large number of sites. Probably you cannot get recordings you can try this approach, but if you get recordings from a new site, you cannot expect that the birds behave in the similar way that the ones that you get the relationship before. So there will be some variation. There are some areas that the same species is vocalizing more often than in other areas. So these methods are full of error. And one important thing here is also that I didn't say, that you can use 2, 3 of these methods at the same time. So you don't need to use only the vocal activity. You can use vocal activity by measuring also the sound level and try to discriminate individuals. Indeed, some of these studies, they have used 2 or 3 of these methods at the same time. So in this way, you are getting this error lower and lower and lower.
Cristian, maybe it's still in the same line of the temporal resolution of the study. So one of the results that you showed was that regarding the coefficient of variation, there was a given amount of days that was better to have a better to have their best estimates. The estimates were more reliable. But I was wondering, considering these possibilities of variation during the season, what's the suggestion? Is it better to have more days, sequential days, or days spread all over a season, for instance?
Yeah, it depends. The same depends on your question. For example, we were only working for the breeding season, but in that graph that I saw for the DuPont slur, the field that I saw much bigger, we also include some data for the post-breeding period, and we didn't show a big difference in the number of recording days. But so I will say that if you need to focus your recordings and your Q rate or the relationship or whatever for a concrete period. If you are using a Q rate during the breeding period, you cannot apply the same Q rate for recordings collected during the winter. So you will, maybe you need to know which is the Q rate of the species during the breeding season, during the non-breeding season, etc. It makes it too much complicated, but usually, probably now with this technique, we are starting to monitor over a year or during different seasons. But today, most of the studies, they focus only in the breeding period. And so we stick to this breeding period for the DuPont's larvae because it's when we have the money to get to the field, to have observers working in the field, etc. But yes, you are. So I cannot say, I would say that it's better if you focus for a concrete period, if you are going to apply some of these methods. Otherwise, if you want, imagine if you only compare the vocal activity over a year, you are not comparing if there are changes in vocal activity or an abundance of the species, you are describing the seasonality of the vocal behavior of these birds. So this is a different question.
Can I follow up on Larissa's question about the coefficient of variation? First, thanks for a really great presentation, Cristian. That was really interesting. It's great stuff. It looked like you had started to make those coefficient of variation like cutoffs for a couple different species. And I am wondering if you think it would be possible to find generic vocal activity rate thresholds that you could apply to multiple species that aren't necessarily the same count, but some statistical cutoff point that would allow you to look at vocal activity rate for species, even if you don't have field data about the number of individuals or the Q rate. So sort of like going down like a dangerous path of trying to estimate these bird abundance without any field data, just from acoustics, if you can look at consistent statistical patterns like that. What do you think?
Yeah, that is the idea of this article, that this is under progress. This is because I put the no Twitter. So this is a study that I'm doing with two colleagues, with three colleagues. One of them is Diego Yusia, that Larissa knows very well. So the idea was the idea is even today to publish like a multi-taxa protocol, because we have also data for frogs and for bats, and of course for birds. That is the one that I saw today. So the idea was to say, okay, if you are going to put the recorder for monitoring frogs and you have no idea about how many days you need to leave the recordings, at least put them for 1 week, because based on our data, with 1 week, you reach a coefficient of variation of 20% for most of the species. So I totally agree, this is very dangerous. I started very happy with that study, but I was starting to think what reviewers will say about this coefficient of variation, because one problem of the coefficient of variation is that, of course, it will be always zero as when you reach the total number of recordings. So there is a great bias with this approach. So if you are recording only 10 days, you will reach always a coefficient of variation lower than 20% with 8 or 9 days. So maybe you are providing monitoring recommendations not very robust, at least for some species. So I was starting to be afraid of that article, and I think this is in my desk. And I don't know if someday I will try to use a different approach and open. So if someone hear what I said and have some ideas, hello, welcome, write to me. But yes, the idea was that, Connor, to do something like that, to provide some monitoring guidelines for birds, frogs, and bats. But I don't know if we will do it or not. At least I can show in this kind of talks to say that there are a lot of difference between species. So you cannot just, if I describe this, you need to record for 9 days to have a low error. Okay, this is valid for the DuPont's lark, but not for a lot of other species. So probably you need to apply a similar protocol for a concrete target species. You can follow our protocol, but you need to estimate the number of days for your species.
So Dimitri has a very cool idea that I'm wondering if either if you want to say a few words about this this thought, or I can try and summarize if you prefer.
Okay, I'm trying to read. I don't know about if there should be a public database about that.
Yeah, of Q rates.
Okay. Yeah, for sure, but I think this is not close to exist because at least for the cure rates of target species, there are very, very few information. Maybe for the vocal activity, it will be a little much easier to collect the data from some published papers, something like that, but maybe just for 20, 30 species, I don't know, but at least I think this is usually provided in several articles, no? That we recorded in 10 days and we detected 1,000 vocalizations. So you can estimate the mean number of vocalization per hour, something like that. There is not a public database, but it could be some kind of meta-analysis or data paper. It could be possible, but for the cure rate of the species, I think there is very, very few information. There are just some few studies that they try to assess the breeding status of some concrete species, if they were breeding or not, if they were mating or incubating, well, not the male, but the breeding status of the species based on the cure rate, because there are some species that they vocalize more often when they are mating. So it was a way to know how many mating birds you have in a population, maybe to separate breeding from not breeding, something like that. But there are very, very few data. And even that assessment that we did, that I saw at the very end, very fast, but the cost effectiveness assessment, how many males or how many times you need to report. I think this is the first assessment trying to give some guidelines about for estimating a reliable cure rate of a species. So it could be perfect, but I think that we are not close to have this database in the following years.
Well, we had a recent presentation last week for our center, and one of the main takeaways was that there's a need in acoustic monitoring to get a better sense of Q rates, or how often a given species vocalizes and over what range those vocalizations are detectable. And so it seems like your work definitely emphasizes that same point. And this would probably be a worthwhile project for someone that's interested in better understanding bird abundance.
Okay. And I will say something that I didn't say in the presentation, but I think that's very important if you want to estimate bird density and not only have an index of bird abundance, is that you need to know the radius of detection of your recorder. Because most of these studies, they applied these methods, but they didn't know if they were detecting birds up to 100 meters, 150, 200, or 500 meters. So in this way, you are not using you don't know the radius. Maybe you can have an estimate of bird abundance and see if this abundance vary over the season or have an impact on the flying activity of honeybees. Of course, you can use them for some questions, but you cannot get a value of bird density. For that, you need to know which is the area that you are recording. So this is something that most of the studies didn't do. Indeed, I didn't do in my first study, the one that I talk about the vocal activity rate in ibis. I didn't have a fixed radius. So I said, I know that we are able to record the European beetle up to 200 meters. So we consider that we are recording up to 200 meters. But this is not true, because at 200 meters, maybe you are able to detect the species, but only 5%, 10% of the vocalization, no? Because there is an imperfect detection when the distance is longer. So this is something that probably most of the studies that they will publish in the following years, you will need to consider this fixed radius, because okay.
Carly, did you have a question, comment?
Got answered in the chat.
Okay, nice.
Okay. Yeah, I'm seeing some comments about this Accurate project. So this study that I talk about the multi-taxa monitoring protocol, this is carried out also with Thiago Marquez, that I think that is one of the leaders of this Accurate project.
Yeah. Okay, everyone. I think that we hit our scheduled time, and once again, thank you, Cristian, for having the time to give us this amazing talk. Congratulations on the effort and putting all of these different techniques together in this review paper. And I hope that we can continue this conversation in these other channels that we have to discuss these topics on passive acoustic monitoring. And yep, I think we'll see you again in 2 weeks.
2 weeks. We've got a very interesting presentation, not about birds, not about anything terrestrial. It'll be our first marine focused acoustic method series in quite some time, and should be very, very, very cool one in 2 weeks. So Leah Buffo, I'm crossing my fingers that I have that very French last name pronounced correctly, but Leah, who is sitting right next to me, will present about a very cool new method to learn about marine mammals using fiber optic cables. So this new way of listening and learning. So thank you, everybody. This is, I think, a record number of people attending. So happy to build this community. Everybody's talking with each other in the chat. It's really cool. So we'll see everybody in 2 weeks' time.
Okay, thank you all.
And
Thank
take care.
you. Bye-bye.
Bye.
Okay, thank you. Bye.
Thank you so much.
Okay, Ben, thank you for organizing
Hey,
this talk
Christian, that was
and for
awesome.
everything. I think
For
that I
sure.
talked too fast some of these methods, but otherwise it is impossible to give an overview in 30 minutes with so much methods.
Much better to leave 20 minutes. That was a really great discussion and question answer session. People were writing in the chat, discussing ideas amongst each other. I mean, you covered a lot, so it was good to leave people with some time.
Okay.
Hey, Destiny.
Hello.
Hi, how are you?
I'm fine. How are you?
Good.
I didn't get the mail early. I didn't get the mail, so a colleague of mine sent me the link to join in, and I joined late. So I was trying to see if I could get a recording, and then maybe you add me to the mail list so that I can join.
Yeah. So your name, I'm just writing your name. If you can send me your email address in the chat.
I wrote it there.
Great. Let me see if I can see it.
That's Deji@gmail.com. Can you see something like that?
Got it. Oh, you sent it before I asked. Nice.
Yeah.
I'll send you an email when it's posted.
Okay. Thank you very much. Yeah.
For sure.
Bye-bye.
I'm grateful. Yeah.
All right. Bye, everybody.
Okay. Bye, Ben. See you.